Hi, I'm Marissa D. Wilson, and today I'm here to discuss the role of cell signaling in epigenetics, or Trotsky, a pathway. Please take a look at these living organisms, from the single-celled eukaryotic yeast to the gram-positive bacteria Staphylococcus aureus, sunflower plant, and even human beings. Cell communication is essential. How do these organisms communicate? They communicate through a process that reflects the complexity of its organisms. As complex as these communications can be, each pathway allows key processes such as the way that yeast mate, bacteria measure its population density, sunflower transport nutrients through its system, and human development. It starts with a signaling cell that wants to send a message and makes an extracellular molecule such as proteins, peptides, amino acids, nucleotides, and steroids that the target cell detects on its receptor. The roles can switch as the target cell responds to the signaling cell. The range that the message travels determines what type of extracellular signal molecule that the signaling cell uses. Now please observe my slide. We have Dion and Cher. Cher calls Dion. Cher is a signaling cell. Dion is a target cell. And the phone that Cher uses is the extracellular molecule, while Dion's phone is a cell receptor. And Dion's response to her, especially as she walks towards her, is the intracellular signaling molecule. So, multicellular organisms can communicate publicly via endocrine cells, which release hormones throughout the organism's body. In plants, this will be done through the plant sap. Human hormones travel through the bloodstream. Next is neuronal signaling, which relies on the quick and specific responders called nerve cells. They can communicate over long distances like endocrine cells, but the difference is that nerve cells communicate through private lines, unlike their public counterparts. The extracellular signal molecules that nerve cells produce are called neurotransmitters. For example, nerve cells can produce acetylcholine, which excites nerve muscle synapses. The more local approach, on the other side, is a process of paracrine signaling, which involves a signaling molecule traveling through extracellular fluid. This allows them to communicate with nearby cells as local mediators, such as histamines, which dilate blood vessels. They also can respond to their own mediators in the process of autocrine signaling, which influences the survival and growth of cancer cells. Lastly, we have contact-dependent signaling, which is direct and has the shortest distance. Instead of secreting a molecule like the other forms, the cells contact signal molecules located within the signal cell's plasma membrane and the target cell's receptor proteins located in its plasma membrane. This can influence development as it tells cells how to differentiate. Extracellular signals get to their target cell with the help of intracellular signaling pathways. They break down into 5 stages: relay, amplify, integrate, distribute, and feedback. Relay involves spreading the signal. Amplification makes the signal stronger. Integration combines the detected signals. Then the signal is distributed to effector proteins, and then the signal receives feedback. The extracellular signal passes to different intracellular signals until it reaches the target cell. The result is the cellular response. Cell response can be fast, slow, or different based on which signal tries to interact with it. These intracellular signaling proteins are vital as they can act like molecular switches for signal pathways. This effect is similar to that of epigenetics. Now, what is epigenetics? Epigenetics is a scientific study of how behavior and environmental factors influence gene expression. Diet, exercise, stress levels, and drug consumption are all factors that determine an organism's instructions for genetic changes. This happens by changing how your body reads your DNA sequence. You see the change in the phenotype while your genetic sequence remains the same. Epigenetics work on the cellular level throughout an organism's development. Whether a gene turns on or off influences the cell's role. For example, if a stem cell will differentiate into a muscle cell or a nerve cell. It also influences which proteins are made and the likelihood of an organism developing illnesses. So as we ponder significant changes, I know what you are thinking. Marissa, are these changes good or bad? The answer is it depends on what environment, behavior, or stage of development triggers the epigenetic change and how long it occurs. For example, if a mother improved her eating habits and does not consume drugs, it will positively affect the development of her child. Her environment, such as exposure to smoking and stress, also plays a role in creating a positive epigenetic change. A negative scenario would be if we have a person who routinely engages in unhealthy habits like Archer, who is pictured on the right. Then they will experience a negative epigenetic change. The same way that habits can be reversible, so are the changes experienced in epigenetics. There are three key processes that result from epigenetics, which can cause an array of effects. These processes are DNA methylation, histone modification, and non-coding RNA. Let's start with DNA methylation, which is the addition of a methyl group to specific places that attach themselves to DNA. The gene cannot interact with or read the DNA, which turns the gene off. For that gene to turn back on, so to speak, it would have to go through the process of demethylation. Next, we have